Suppressing viral infections or halting their replication is one of the most challenging tasks in medical science. The reason behind this difficulty is the ability of viruses to quickly adapt to any defense mechanism introduced against them. Imagine you've developed an antiviral drug. The virus might just alter its amino acid sequence to bypass the drug's action and continue multiplying in peace. Similarly, if you've created a vaccine, the virus may mutate the relevant epitope, the part of the virus that the immune system recognizes, rendering the vaccine less effective or even useless. Although potent antiviral drugs exist and are used to treat various viral infections, the potential for viruses to develop resistance to these drugs cannot be overlooked. Despite this constant viral evolution, one critical vulnerability of viruses remains: their reliance on the host's cellular machinery. Without access to the resources and processes within the host cell, viruses cannot survive or replicate. This dependency offers another potential approach for antiviral therapy, creating a hostile environment within the host cell that impedes viral replication. Picture living with a roommate who constantly piles up dirty dishes in the sink and leaves stuff everywhere. Over time, the living conditions would become unbearable. Similarly, by manipulating essential cellular factors, we can create an unfavorable environment for the virus, limiting its ability to thrive and spread. In our research group, led by Professor Dr. Eva Herker, we investigate the interaction between human pathogenic flaviviruses and host cell lipid metabolic processes. These flaviviruses include Zika virus, ZIKV, West Nile virus, WNV, tick-borne encephalitis virus, TBEV, and dengue virus, DNV, all of which are transmitted by arthropods, as well as the hepatitis C virus, HCV. Numerous studies have shown that these enveloped, positive-strand RNA viruses are highly dependent on a variety of host cellular lipids for their replication. This reliance on lipid metabolism led us to explore the potential role of lipid-synthesizing enzymes in viral replication. As part of our work with the Loew Center Druid, novel drug targets against poverty-related and neglected tropical infectious diseases, we sought to identify potential antiviral targets by focusing on the lipid metabolic pathways within host cells. To do this, we used RNA interference, RNAi, to selectively deplete key enzymes in different lipid synthesis pathways and assess their impact on viral replication. Our initial experiments were conducted using human hepatoma cells, HA7, which are permissive to the viruses we study, DNV, WNV, TBV, and ZIKV. Almost immediately, it became apparent that neutral lipid-synthesizing enzymes, which produce triglycerides and cholesterol esters, play an important role in orthoflavorous replication. Neutral lipids are stored within organelles called lipid droplets, which act as energy reservoirs and are utilized by viruses to support their replication. Our experiments showed that when we depleted the enzymes responsible for triglyceride synthesis, namely diacylglycerol oacyltransferase, DGAT, 1 and 2, the infection rates of WNV, DNV, and ZIKV were significantly reduced in knockdown cells. Interestingly, while 3 viruses showed lower infection rates, only ZIKV demonstrated a corresponding decrease in viral titers, suggesting that ZIKV might rely more heavily on triglycerides than the other viruses. When we knocked down the enzyme sterol oacyltransferase, SOAT1, which is responsible for cholesterol esterification, we observed a broader effect. SOAT1 depletion resulted in lower infection rates and viral titers for WNV, TBV, DNV, and ZIKV. This indicated that cholesterol esterification is essential for the replication of these viruses, particularly ZIKV, which exhibited the strongest response. Based on these findings, we decided to focus our efforts on understanding the role of DGAT1/2 and SOAT1 in ZIKV infection specifically. The results from our enzyme knockdown experiments, we next explored whether pharmacological inhibition of these enzymes could similarly reduce ZIKV infection. Previous studies had suggested that DGAT1 activity was critical for ZIKV replication in placenta models, so we anticipated a strong effect when using DGAT1 inhibitors in our hepatoma cell cultures. To our surprise, ZIKV continued to replicate even in the presence of DGAT1 inhibitors, leaving us initially frustrated. The story didn't end there. During her postdoctoral work in the US, Eva discovered that DGAT1 interacts with the nucleocapsid protein of hepatitis C virus, HCV, and this interaction is essential for efficient viral particle production. We hypothesized that a similar interaction might occur between DGAT1 and the ZIKV capsid protein, even if DGAT1 activity itself was not required for ZIKV replication. When we examined this interaction, we found that DGAT1 and the ZIKV capsid protein indeed co-localized and interacted, providing a plausible explanation for why DGAT1 activity was not essential, but its presence was still important for ZIKV infection. On the other hand, SOAT1 inhibition initially yielded mixed results. When we looked at viral titers, the data were inconsistent, showing only slight reductions with one of the two SOAT1 inhibitors we tested. However, a more detailed analysis revealed that SOAT1 inhibition led to a dose-dependent reduction in viral protein levels and less severe cytopathic effects in infected cells. ZIKV primarily infects neural tissues, so we decided to repeat our experiments in neural cell lines to see if SOAT1 inhibition would have a stronger effect. This decision proved to be pivotal. In contrast to the minimal effect seen in H7 cells, SOAT1 inhibition significantly reduced ZIKV infection in microglia and astrocytoma cells, which are key target cells in ZIKV infections. Encouraged by these results, we extended our experiments to more sophisticated models, including 3D cerebral organoids, a model developed by one of our colleagues, Vinicius. In this complex system, although it's exciting to observe that a substance can stop viral replication, the next question for any scientist is always, why? To answer this, we systematically examined the stages of ZIKV replication affected by SOAT1 inhibition. We found that while viral entry and RNA replication were unaffected, the production of viral proteins was significantly reduced. This suggested that SOAT1 inhibition was disrupting viral morphogenesis. We then asked the key question, how does inhibiting SOAT1 block viral replication? SOAT1 is responsible for converting free cholesterol into cholesterol esters, so its inhibition results in an accumulation of free cholesterol within the cell. Could this buildup of cholesterol be detrimental to the virus? To test this, we supplemented SOAT1 inhibitor-treated cells with cholesterol esters and observed no rescue of ZIKV infection. However, when we added cholesterol itself, ZIKV infection was significantly reduced. In summary, our research showed that inhibiting the cholesterol esterifying enzyme SOAT1 reduces ZIKV infection by creating an imbalance between free cholesterol and cholesterol esters. This imbalance likely disrupts the lipid environment necessary for efficient virus particle morphogenesis, leading to fewer infectious viral particles being produced. By targeting essential host cell factors such as SOAT1, we can potentially develop novel antiviral therapies that limit the ability of viruses like ZIKV to replicate and spread.